T cells do what? Stop that hyperactivity of the immune system. That's why it is called regulatory. Okay, inhibiting your immune system, right? From what? Inhibiting from acting against your healthy cells. That's how these T cells, regulatory T cells or suppressor T cells, inhibit autoimmune disease or prevent autoimmune disease. Stops hyperactivity of your immune system. Is it clear?
So is that what like you get medicine for? Is that what is in the medicine?
Medicines can activate the suppressor T cells. Yes.
Okay.
Yeah. Make sense?
Mm-hmm.
So it reduces the hypersensitivity or hyperactivity of the immune system. Yes. That is one way medicine works. So those are different types of T cells. Now, antibodies. Antibodies are proteins. Immunoglobulins. You remember albumins, globulins, fibrinogen. So globulins are antibodies. At that time we have learned it, right? Globulins are antibodies. So those globulins are called immunoglobulins because they give immunity. Is it clear? Immunoglobulins. And these immunoglobulins are gamma type globulin portion of blood. Gamma. Secreted mainly, we'll say, secreted mainly by plasma cells. Some B cells are converted to plasma cells and they produce plenty of antibodies. We talked about that. And antibodies bind with the antigens. That's why they are antibodies. Antigen, antibody complex. So, if this is an antigen, you must remember a few minutes ago I told you there are harmful sites on the antigen. So, if I think these are the harmful sites, antibodies do what? Antibodies usually look like T or Y, like this. Okay? Y shape or T shape. So what will happen? This antibody will get attached to the antigen. This one will come here, will get attached to the antigen. Okay, so this is antibody and this is antigen. So you see the antibodies are blocking the harmful sites. So this antigen is now harmless, cannot cause harm. But you see here that antibodies must get attached to the antigen to inactivate or kill them. And this is now called antigen antibody complex. Very simple. Together, antigen antibody complex. So by forming complex, antibodies inactivate or destroy what? The antigens. Is it clear? Antibodies or gamma globulins are usually T or Y shaped monomer or 4 looping linked polypeptide chain. So how many chains? 4. 2 chains are called heavy chains and 2 are called light chains or H and L chains. So if you see the structure of an antibody, you see it looks like an Y or T and it has 2 larger or heavy chains or H chains, 2 lighter or smaller chains. But one thing you notice here, they are linked by disulfide bonds, those yellow bonds, disulfide bonds. Now, one thing you notice here, these are heavy chains and these are light chains, okay? Both heavy and light chains have two parts. One part is called constant or fixed part. For example, this part of this one is fixed or constant, and this part is variable. So each of those 4 chains has constant and variable part. Same way, the light chain, let me change the color. This is variable part, this is constant part, okay? Same thing here, variable and constant, okay? So like this, look at this. These are the chains, okay? 2 parts, make sense? This part is constant, fixed. This is variable, okay? So variable part can be what? Changed, make sense? This part can be changed, okay? Which is very good, because you see, by only changing this part, I can make this antibody work on different types of antigens. Because this part, variable part, gets attached to the antigen, right? The variable part, you see, this part gets attached to the antigen. So now, if one type of antigen is here, like, this is the antigen, okay? So this is antibody, okay? So this red will only, be able to destroy this one or inactivate this one. So this antibody now got red variable part, make sense? So this one will be able to work on this. Does it make sense? Same color, just think about that way. Okay. Now if the antigen is this type, this variable part will be changed. This part is remaining same, right? Constant part. This part, the antibody is changing only this part, right? So it will now work on this antigen. Make sense? So you see, only by changing the variable part, the antibody can work on different, specific antigens. Okay? So which is efficient way, right? Instead of changing the whole antibody, part of it is being changed, right? So it will work on different antigens by changing the variable part, which is more efficient way to work. Okay, now 3 mechanisms are most commonly used to destroy the antigens. One is called neutralization, another is called agglutination, and another is precipitation. So when antibody complex is formed, either of those 3 things will happen. So antigen antibody complex is the first thing that must happen, and then that complex can work by neutralization, neutralizing the antigens, or by forming agglutination that you have seen when you did the blood typing, that clot is formed. So that is agglutination and precipitation. So here you see, I showed you these are the harmful sites of this antigen, right? Remember, these are the harmful sites, right? So these antibodies are blocking or masking the harmful sites, right? And making the antigens harmless, okay? inactive. So this is neutralization. This process is called neutralization. Neutralizing, inactivating. Now, sometime after the antigen-antibody complex is formed, that whole complex become immobilized, cannot move because of weight. Okay, so antibodies will get attached, and that antigen's movement will be restricted. Okay, so that is another process that is called precipitation. You know, if something is, weight is heavy, then it will not be able to move, right? So it will stay somewhere, precipitate somewhere. So that is another method. And agglutination is clamping or clot formation. Another way you can say clot formation. Agglutination you have already seen when you did the blood typing. You know, you saw the clot formation, right? When you mix the antibody on the red blood cell, you have antigen, right? In the blood you have antigen, and you drop that serum. Remember that? And then agglutination occurs. That means clamping or clot formation occurs. So those are three common ways. You see the picture: neutralization, agglutination with red blood cells, precipitation. So basically, if you see the agglutination and precipitation, many antigens and many antibodies are chained together. So basically, antibodies are chaining the antigens, tying them together, right? So that structure gets heavier, cannot move, or clot formation. Now, antigen-antibody complex can release a chemical that is called complement. So that complex, this is the complex, can release a chemical that is called complement. This complement is activated by a number of reactions. We are not going to talk about all those reactions, but through a number of reactions, the complements get activated. Okay, and then the activated complements can do what? Can enhance phagocytosis, enhance inflammation, and can cause lysis of the cell. First, let's see. Complement can activate phagocytic cells. So, increase phagocytosis or cause phagocytosis. Make sense? That is number one. Number two, can enhance inflammatory action. Inflammation, which is very important. Now you tell me, you have already learned inflammation, right? So, when inflammation occurs, at that site some chemicals are released, right? Remember, that causes vasodilation, right? More blood goes there. If more blood goes there, you got inflammation here, okay? More blood flows there, right? We know that already. That's why that redness occurs, right? So if more blood goes there, more white blood cells will go there, right? More antibodies will go there. We know that. So immune response in that area will be more or less? More, because more white blood cells are going, more antibodies are going there, right? So that area will have more immune activity. Make sense? So, and inflammation is very helpful. It helps immunity in that area. Okay. That's why inflammation occurs. And then lysis of bacteria or virus. What happens? Complements work that I have actually explained a little bit when we talk about T cells. This is bacteria or virus. For example, this is a bacteria. The complement increases the permeability of the bacterial cell membrane. So when the permeability is increased, more water influx occurs. So water from outside enters into the bacteria. Okay. So complement does what? Increases the permeability, right? So through the cell membrane of bacteria, water will easily get in. Make sense? So more and more water will get in, and the bacteria will get bigger, and at one point what will happen? Burst, lysis. Make sense? So that's how the complement works. 3 ways: increasing phagocytosis, enhancing inflammation, and increasing the permeability of the membrane of bacteria or virus, okay, and destroy them by lysis. Make sense? Got some idea? You will learn more in microbiology. They will explain better. Okay.